Assalamu alaikum everyone. I am Mehwish and the topic I am going to present is about monoclonal antibody. So before going deep in the topic, I will explain what we are going to study in this presentation. In this presentation, I'll be explaining what is monoclonal antibody, what is antibody, the difference between polyclonal antibody versus monoclonal antibody, principle of monoclonal antibody, Hybirdoma technology, procedure of the Hybirdoma technology, I will explain the whole procedure of this technology and why half medium is used and the therapeutic use of the monoclonal antibody. Now, what is monoclonal antibodies? As we all know that antibodies, what are antibodies? They are basically produced by our body to defend our body against different kind of germs. When germs act in our body, our immune system naturally produces antibodies that basically kills those germs or particular type of foreign particles. And what are monoclonal antibodies? By definition, antibodies derived from a single clone of plasma cells, all having the same antigen specificity produced against a single epitope of same antigen. What does this mean? Like we all know that antibodies, they are produced from the B cells. Now here I have written the plasma cells. Now plasma cells are the modified version of the B cells. So in case of monoclonal antibodies, all the antibodies which are derived from a single clone of plasma cells, and that antibody, it is specific for a specific type of antigen. Like there are multiple type of antigens, and you heard that the monoclonal antibodies, they bind to a very specific kind of antigens. So in short, monoclonal antibody produce specific type of antibody which is specific for a specific type of the antigen. So before going deep into the topic of monoclonal antibody, first I'll give you a very brief intro of what is antibody. Antibodies, they are the immune system related proteins which are called immunoglobulins. We call antibody as immunoglobulins. Each antibody consists of 4 polypeptide chain, 2 heavy and 2 light chain, and they're joined to form a Y-shaped molecule. If you see here, the figure 10 points this, which is this is an antibody structure. There, it's a Y-shaped structure. It's having a light chain, a heavy chain, and there's an antigen binding site. To that side, the antigen binds, as I also mentioned here in this diagram, the antigen is, here is an antigen, and these antigens are binding to, the antibodies, they bind to a specific kind of antigen. Now, antibody is a protein which is produced by immune system in response to the presence of antigen. The difference between, we have heard about the monoclonal versus polyclonal antibody. We have heard there are polyclonal antibody, there are monoclonal antibody. So what is the difference between both of them? So polyclonal antibodies are the antibodies which is arise from multiple clone B cell. Like different kind of B cell are producing different kind of antibodies, and that antibodies are not specific for a specific kind of antigen. They are specific for many kind of antigens. But in case of monoclonal antibody, they are the antibody from a single pairing clone. Like in short, we produce those and monoclonal antibody. Now, the second point is that there is a mixture of antibody that detect the different epitope on an antigen, like different epitope on antigen. And third point is that the typical in serum in response to antigen. It means that in the polyclonal antibodies, they are generated all the time in response to antigen. It's not specific. It is present in our body, and our body is continuously generating it. And what is monoclonal? And in case of monoclonal antibody from one clone, it detects only one epitope of an antigen, just specific. Okay? And it's usually produced in laboratory using the Hybirdoma technology. It needs to be produced. It's a very specific interaction. Here the point, epitope is discussed. What is epitope? So antigens are having multiple epitopes which enters the body. Now, each epitope, it may stimulate one clone of B cell producing one type of antibody. So when only one clone of B cell, it is stimulated by a single epitope of an antigen, then only monoclonal antibodies are produced. Here in the diagram, we can see the antigens. In monoclonal antibody, antigen is binding to a very specific epitope on antigen. While polyclonal antibodies polyclonal antibodies, they are binding to different epitopes on antigen at the same time. So now, what is the principle of monoclonal antibody production? Well, the principle that helps us to produce monoclonal antibody is known as Hybirdoma technology. With the help of Hybirdoma technology, we can generate the monoclonal antibody. As monoclonal antibody is kind of laboratory antibodies, it's laboratory made antibody, sorry. So what is the principle? How we basically do now, the clone of B cell. The first step is that a clone of B cells stimulated against a single epitope of an antigen. So it is fused with an immortal cell, like a myeloma cell, which is capable of multiplying indefinitely to produce hybridoma cell. Now, what does this mean? That whenever in monoclonal antibody, we basically want to produce antibody for a specific antigen of our desired antigen. We want to produce any antibody for desired antigen. So the first step is that we will stimulate the B cell. B cell is responsible for making antibody. We'll stimulate the B cell against that a specific antigen we want to produce antibodies. And after, when it is stimulated, when it is stimulated, when the B cell is stimulated with a specific kind of antigen, that stimulated B cell is then fused with any immortal cell. Now, what is immortal cell? That cannot die, that can live. Like immortal cell, like myeloma cells, which is capable of multiplying indefinitely to produce hybridoma cell. Hybridoma cell is also known as fused cell. Now, this hybridoma cell has two unique properties. Like they will produce monoclonal antibody of same antigen specificity due to B cell component and multiplies indefinitely, producing a clone of identical cell due to immortal cell components. Now, I will be explaining this principle deep in a next slides. So this is the whole principle. We will take a clone of B cell, stimulate with specific antigen. After when it gets stimulated, we will fuse it with an immortal cell. After fusion, a hybridoma is produced. Now that hybridoma is responsible to produce a monoclonal antibody of that same antigen specificity, and plus it will multiply indefinitely, producing a clone of identical cell. Hence, we will get more monoclonal antibodies. Here, this is an overview regarding to the Hybirdoma technology. The overview is like that. In the first step, so the general overview of the Hybirdoma procedure is like this, like the mouse. The mouse, it is immunized by a specific antigen injection against which monoclonal antibodies have to be produced. After 72 hours of immunization, spleen is collected from a mouse. Spleen cells, they are collected from a mouse. Okay, why spleen cells? Because we know that in spleen cells, there will be a B cell, sorry, not B cell. There will be an antibodies as well as antigen, both are present in the spleen cells. So we'll collect the spleen cells from the mouse, from the spleen cells are what? They're the antibody producing B cells. Now, that B cell, it will be fused with the myeloma cells. Myeloma cells are what? They're the cancerous cells. They will be fused with the B cells. Now, the results is like not that came all the B cell will be fused with myeloma cell? No. The fusion outcome of the fusion include now they both the B cells and the myeloma cells they may fuse and they cannot fuse. So the possibilities is this. Now, to as we all know that after fusion, we will select the fused cell that can produce the monoclonal antibody. How it will be done? How exactly it will be done? It will be done using culture in a hat medium. Using culture in a hat medium. We will use a hat medium. We will use a hat medium. Sorry for the bad drawing, or you can say we can use. I will use a razor here. So we will do culture in a hat medium. Why culture in a hat medium? Because we want to extract our fused Hybirdoma cell, and that can be only possible using the hat medium. Purification is done through hat medium. So as we can see, not all the cells they are not all the cells are fused on. For the possibility of fusion is very like 1 to 2%, and then which can produce a monoclonal antibodies. So how the whole process? This is a general overview. This is a general overview of Hybirdoma technology. I will explain this whole procedure in a brief way in our next slides. So, okay, one more point here is that the fusion, that here, the fusion, it occur in the presence of, okay, it's not working, in the presence of PEG. I'm sorry for the interruption here. Just generally an overview. Let's go to a brief concept of the topic. This is the whole procedure. Now, what is the procedure of the Hybirdoma technique? The first of all, mouse splenic B cells. As I explained, that mouse is injected with an antigen containing the desired epitope. Okay? You are injecting the mouse with an antigen against which a specific antibody will be produced. After an interval around 72 hours, the mouse splenic B cells are obtained, which would contain the mixture of B cells activated against the epitope of antigen injector. Like we will extract the splenic B cell, and that splenic B cell, we know that it contains a mixture of B cell, which is already activated, it means stimulated against the epitope of the antigen injected. Now, why we use myeloma cell? This mouse splenic B cell, it's all about the B cells. Like we are activating the B cells with the help of specific antigen. Then we use myeloma cells. In Hybirdoma technique, we use myeloma cells. Why we use myeloma cells? Because they are used as a source of immortal cell. Myeloma cells are, what are myeloma cells? They are basically cancerous plasma cells, closely resembling mouse B cells, hence are compatible for the fusion. They're the cancerous plasma cell and they closely resembles the mouse B cells, hence they're compatible for fusion. Like fusion is kind of very difficult. It's not a very easy process. So the question arises here that why we use myeloma cells? Because we found out that myeloma and the spleen cells, they are structurally similar so that the spleen cells, so that they can easily fuse to each other. Generally, fusion of cell is not that much easy, but as they both are compatible to each other, that's why we fuse the mouse splenic B cell with the myeloma cells. इसके अलावा, the myeloma cell have capacity to produce their own antibodies. Hence, own antibody. This is the specificity of the myeloma cells. It can produce its own antibody. It basically resembles to that of the B cells, splenic B cells, and it's as used as a source of immortal cells. So that's why we fuse myeloma cells with the mouse splenic B cell. Now to use the myeloma cells are genetically modified with two mutations. And that mutation is known as double mutated myeloma cell. Why? Why we mutate the myeloma cell? Why? Because they lose the ability to produce their own antibody, but retain the immortal property. So for the fusion, we basically mutate the myeloma cells. Why? To lose so that they lose their ability to produce their own antibody, but retain immortal property. So in short, this is the first procedure of the Hybirdoma technique. Like first we activate the mouse, first we activate the splenic B cells, and then we those act against the specific antigen, and then those activated splenic B cells are then again fused with the myeloma cells, which we by ourselves first mutate them so that they lose their ability to produce their own antibody but retain immortal property. Why we do this? Why? What is the main reason behind it? So that I'll be explaining this in the next slide. Now, how fusion occurs? Okay, fusion of B cells, which is HGPRT positive, and myeloma cell, which is an HGPRT negative. How the fusion occur between both of them? Now, fusion. The mouse cleaning after selection, first we go for the selection. We choose the we stimulated B cell, we stimulate by ourselves, then we go for the myeloma cell, we by myself mutate them and both are compatible. The next step in Hybirdoma technology is to fuse them. Now, how the fusion occurred? Fusion occurred. The mouse splenic B cells and mutated myeloma cells, they are fused in polyethylene glycol bronze. The reaction chamber, now three types of cell can be generated. When B cells, it gets fused with the myeloma cells, you can get three different kinds of cells. It may be, it may be, wait, it may be an unfused myeloma cell, it may be an unfused myeloma cell, unfused myeloma cell, it may be an unfused mouse splenic B cells, it may be an unfused mouse splenic B cell and a fused hybridoma cells. Hybridoma cells, hybrid. It is in a hybrid of B cells and the myeloma cell. So after this is the hybrid cell. So after, when the hybridization occurred, so after this is the hybrid cell. So when now, three different kind of cells are generated, and we don't know that which one is our fused hybridoma cells. We'll go the next step in the Hybirdoma technology is the purification of the hybridoma cell. How the purification occurs? By purification occurred by the subculturing on a hard medium. The next step is to remove the unwanted cell and to propagate the clone of the hybridoma cells. This is carried out by subculturing the cell in a reaction chamber onto a special medium which is called hard medium. Now, the hard medium is used to extract or separate your hybrid cells. Now, what is hard medium? How it is used and how it is used to separate those hybrid cell from the rest of the cell? It's in the next slides. Let's go to the next slides. What is hard medium? What is hard medium? Or why hard medium is used? Now, hard medium contains an hypoxanthine. It stands for an hypoxanthine. A stands for aminoterrin, and T stands for thymidine. Let's go to a little bit deep in the, or let's try to understand the process in detail. Now, purine synthesis in mammalian, it's a very like you can say it's a very simple point which we need to explain, or how we will separate hybrid cell or from normal B cells of spleen and a myeloma cell. For that, we will need to understand or we need a hard medium. So let's read a little bit backdrop of the HAP. Purine synthesis in mammalian cell, for example, spleen cell. It occurs by either de novo or salvage pathway. Like the purine synthesis in mammalian cell, it occurs by either de novo or salvage pathway. Now, a minor which is present in the heart medium, it blocks the de novo pathway. So that cells has to perform the salvage pathway to synthesize purine for its survival. Now, that salvage pathway requires two very important enzymes. One is an HGPRT, which is the known as in hypoxanthine guanine phosphoribosyl transferase, and a thymidine enzyme. These two enzymes are very necessary for a salvage pathway. So any cell that lacks the HGPRT or a thymidine kinase, it cannot grow on a heart medium. It will not grow, it will eventually die. And the example for that cell is myeloma cell. Myeloma cells cannot grow on heart medium because it lacks the HGPRT enzyme. If you go in a bag, here you can see HGPRT minus. It means that it lacks the HGPRT enzyme. It means if we grow this heart medium, if we grow this myeloma cells on a heart medium, it will never grow. These cells can grow, but why we cannot use individually these cells? I'll explain this. Let's have a look why. Fate of cells in that medium, like what is the whole procedure? Fate of cells. Unfused splenic B cells. Like this is an unfused splenic B cells. Okay. This is an unfused splenic B cells. It can grow, but it do not survive long as they are not immortal. When you grow this on heart medium, it has its own enzyme. It has an GPRT that is required for salvage pathway. It will do its salvage pathway, produce purine, but it will not survive as long as they are not immortal. Then the next one is unfused myeloma cells. These unfused myeloma cells, it's clear that it cannot grow as they lack HGPRC enzyme to perform the salvage pathway of a purine synthesis. It definitely cannot grow. It definitely cannot grow. Now, the one that is left, it definitely cannot grow. It definitely cannot grow. This can also not grow because they are immortal, like they're not immortal, so they will eventually die. The only point which is left the only cell which is left, it's known as Hybirdoma cells. They can grow and survive long. As Hybirdoma, why they can grow and survive long? Because it's in a hybrid. It's a hybrid of B cells and it's a hybrid of myeloma cell. Myeloma cell has the potential of immortality, while the B cells, they have those particular enzymes that is required for the growth. So they will eventually grow on a hard medium, and as a result, we can have those, and as a result, it will extract or purified our hybrid cell or hybrid cell from the rest of the cells. This is the fate of cells in a hard medium. What is the next procedures, which is Hybirdoma technique? Now, what is now? This is all about the Hybirdoma technology. Like we just, first of all, we stimulate the B cells with a specific antigen. Then we inject that antigen into a mouse splenic mouse, and we extract that mouse splenic cell because it is now activated and contains the B cells as well as the antibody as well as the antigen. And after that, we extract the spinning B cell, we fuse them with myeloma cells. The myeloma cells, we do a little bit mutation on the myeloma cell, like so that they does not produce an antibody, but they have the retain the potential of immortality. And after the fusion, we go for a purification. Why? Because we know that we just need our specific hybrid cell. Only the hybrid cell has the capacity to grow the heart medium. Then what is heart medium? I explained the HAT medium that is containing hypoxanthine, aminoterrane, and thymidine, and what is the purine synthesis phenomena, and why we use the HAT medium. It's all explained very briefly. Now, have we select a particular hybrid cell that contains that will produce our monoclonal antibody for that specific antigen, because we need our own. It's a different kind of a different kind of hybrid cell will be produced. It's not only all will having particular efficiency or producing their antibody, but we need the hybrid cell that is specific for. We need the hybrid cell that is specific for a specific kind of antigens. So that they can produce a specific monoclonal antibody for our desired antigen. So now the selection of individual hybridoma cells, how we will select this. If the original antigen used as a multiple epitope, many B cell would fuse with myeloma cell, definitely, to produce a mixture of hybridoma cells, each having specificity for one epitope. So at the very beginning, we give the antigen, so basically to stimulate the B cells. Now, to get the specific monoclonal antibody for a specific antigen, we will use a very specific epitope. The antigen epitope must be specified so that it should not produce multiple antibody. Now, we will try to separate cell in such a way that we get a particular individual hybridoma cell. How? Now, medium containing hybridoma cell is then diluted into a multi-well plate to such an extent that each well contains only one cell. And hybridoma cells producing the desired monoclonal antibody, they are selected by a radioimmunoassay ELISA technique. These are technique which is using the specific antigen fragments that are selectively proliferated. Now we will try to separate cell in such a way that each well gets only one cell. So this is how the whole procedure is explained. If we see here, like we can see here the antigen is injected into a mouse. Then we will extract the spleen cells. Sorry, we will extract the spleen cell and look at this. We injected a mouse with a particular antigen. We'll extract the spleen cells, and after that, we will fuse we will fuse the spleen cell with the myeloma cells, the cultured myeloma cell line. We will fuse both. We fuse now in the presence of polyethylene glycol. After fusion, we will go for a separation in a HAT medium, and on like a different we'll grow in the whole HAT procedure will be done. After that, medium containing hybridoma cell is then diluted into a multi-well plates to such an extent that each well contain only one cells. And then hybridoma cells producing the desired monoclonal antibodies, they are then selected by now. How are desired hybridoma cells will be selected? They're selected by the radioimmunoassay ELISA technique. Like on HAT medium, we will separate the hybridoma cells. Then each hybridoma cell is then diluted into a multi-well plate to such an extent that each well contain only one cell, and then particular hybridoma cells of desired monoclonal antibodies are then selected by the ELISA technique or radioimmunoassay using that specific antigen fragment. Why? Because those fragments, your particular antibody will bind to it, and ultimately will get to know, okay, fine, this hybridoma cell is producing this monoclonal antibody, and this monoclonal antibody is bind to a specific antigen, and this is how your antibody will be. You will select your particular hybridoma cells, and then we will culture it, and a lot of antibodies, which are monoclonal antibodies, that we produced. Now, this is all about the hybridoma technology, or sorry, hybridoma technique, and the procedure. I explain how it's produced, the difference. I give the overview. Let's go for a next slide, let's go for a next one. Therapeutic application of monoclonal antibody. Therapeutic. Why monoclonal antibodies are important? Like monoclonal antibodies is being used nowadays for almost every single kind of serological test and a reaction, if you want to, and reaction. If you want to find out specific antigen-antibody interaction, the monoclonal antibodies is really important and really specific and also help us to find out. Now, therapeutic use include treatment of various inflammatory and allergic diseases and cancer. The mechanism by which monoclonal antibody work as therapeutic is to provide, is done by suppress the immune system. Now, why? The suppressive immune system is required in autoimmune diseases like rheumatoid arthritis and many other cases. Kill or inhibit the malignant cells, like it target the cancer cells and help in destroying them. We can mask specific receptors so that these receptors may not get hyperactivated by a ligand that can trigger the cancer formation. So inhibition of cancer can also be done, and inhibition or inhibit angiogenesis. This is all a monoclonal antibody can done. And thank you very much. And this is all about the monoclonal antibody.